Welcome to this new episode of the Virulent Vortex, a podcast in which we discuss infectious diseases across scales, from molecules and pathogens to populations, pandemics, and everything in between. My name is Jaap de Rode. I'm a biology professor here at Emory University and today's host of this show. And today we are joined by Associate Professor of Biology, Dave Civitello. Dave, welcome to the show.
Hi, Jaap. Thanks for having me.
It's great having you here. And you're a biologist, you're an ecologist, so a lot of your work really focuses on disease ecology. So before we dive into your research, can you just talk a little bit about what that means to you to be a disease ecologist?
Yeah, that's a great question. When I teach ecology, I tell students it's all about the distribution and abundance of organisms. Mm-hmm. And so as a disease ecologist, I focus on the distribution and abundance of parasites and their hosts. Mostly I've focused on distributions of parasites across populations, so maybe different sites or different habitat types.
When you say populations, that can be populations of humans, but it can also be populations of water fleas that you have worked on before, but also populations of snails. And I think, a lot of your work focuses on snails and the parasites that are important for them and humans.
Yeah, that's right. So a lot of our work focuses on human schistosomes, which are trematodes, or flatworm parasites. And these obligately, or they must cycle between snails as their first host and humans as their definitive host. And definitive host meaning that they reproduce sexually inside humans, but they also kind of clone themselves inside snails and release new sort of juvenile life stages back into the water where humans can contact them and become infected.
Okay, so these are snails that live in the water. They get the parasites. How do they get the parasites?
So yeah, the life cycle is pretty interesting. So there are two hosts. If you start with adult schistosomes inside humans, these adults will lay eggs inside our bodies. And some of those eggs, about half, will be excreted, depending on the species, either in our urine or our feces.
Okay.
When that excrement ends up in a freshwater habitat, those eggs can hatch.
Yeah.
And when they do hatch, the first juvenile life stage of the parasite emerges, swims through the water, and tries to burrow through the skin of a snail. If it's successful, about a month later, it will have cloned itself many thousands of times, and it will then send out the second juvenile life stage back into the water. Which will try to burrow through our skin. And so this is an infection that's acquired by humans when they contact water. It's not about drinking water. It's not about eating anything. It's really about dermal contact with the water. So that can happen through swimming, through bathing, through washing clothes, fishing activities, even activities like fetching water to wash cars.
So how big a problem is schistosomiasis at the moment?
Yeah, it ranks among the top 3 to 5 infectious diseases in terms of global human morbidity. Schistosomiasis is the proper schistosomiasis disease or acute symptoms are relatively rare. Its pathology is intensity dependent. So the more times an individual becomes infected, the more worms a human is infected with, the worse their symptoms. Many people throughout the world have light infections, and few individuals have really, really heavy infections that cause severe pathology and sometimes even death. So the death toll of schistosomes is not exceedingly high, but the morbidity that they incur on humans is very high, with about 200 to 250 million people infected at any given time. I've met children who show signs consistent with heavy schistosome infections, whose parents indicate they haven't been able to go to school. And so there's a lot of sort of knock-on effects, a lot of other effects aside from the direct pathology.
Okay, so you are not a medical doctor, you're an ecologist. So let's get you back to the ecology and some of the projects that you work on. I think one of the things that you study is schistosome hybrids.
Yeah, so there's been a long recognition that some of these species of schistosomes can hybridize. And I should back up and say that there are about a dozen species of schistosomes that are medically or relevant from a veterinary or wildlife perspective as well. And
can I just interrupt you and ask you what you do mean with hybridize? What does that mean?
Yeah. So this would be interbreeding between two otherwise what were thought to be otherwise separate parental species or lineages, right? So
like a horse and a donkey making a mule, is that a hybrid?
Yeah, that would be a hybrid. Yeah, that's a good example. Schistosomes have kind of classically been recognized by which vertebrate species they infect, and so I'll give you an example. Schistosoma mansoni is sort of the human schistosome that causes intestinal schistosomiasis.
Okay.
Schistosoma hematobium is a human schistosome that causes urinary schistosomiasis. Schistosoma bovis, as the species name kind of suggests, is a cattle schistosome. And for a long time, it's been recognized that Schistosoma bovis and Schistosoma hematobium can hybridize.
Okay.
And species like Schistosoma mansoni can hybridize with other schistosome species as well. So the epidemiological concern is that hybridization can confer, or hybrids could express traits that neither parental species could express. And if those traits include, increased host range or increased virulence, or certain other factors related to transmissibility, this could be problematic.
Okay. So you're saying that through the process of hybridization, you can have two species of schistosomes, and they create new hybrids that may be more virulent, more dangerous, they may spread better, or they may even infect different new animals? Or
Yeah, exactly. We don't know a ton about their distribution or abundance, right? The basic ecology of hybrids. And so a few years ago, we started on a project to investigate the abundance, prevalence of schistosome hybrids in a region of Tanzania where we've got another project that we've worked on for a little while. And so this is an agricultural landscape where humans and cattle share access to the same water bodies. We found thousands and thousands of schistosome infections. And we're now really nailing down the methods, which have been well refined over the last couple of years, last 2 years really, to document whether or not we have hybrids. And funnily enough, we're not finding hybrids.
Interesting.
Yeah. But at the same time, this project has opened up a lot of really neat avenues. And then we started working on and thinking more about cattle. And hybrids aren't the only thing they contribute to the water bodies or to the system, really. So the way that cattle introduce parasites at all to these water bodies is when they defecate. Some of these water bodies are visited by hundreds or thousands of cattle every day, and they're not very big, maybe 10 meters by 20 meters or 10 meters by 50 meters, about half a meter deep. So these are relatively small water bodies that can become loaded with cattle manure.
Right.
And that may sound unappetizing to you or me. It does. But for snails, that's fertilizer for the type of food that they love to eat. So they love to eat algae and bacteria and fungi, other microbes growing on this manure. They like to eat some of the undigested plant material that's in the manure. That is a huge fertilization event that happens day in and day out in some of these water bodies. And so that's really become kind of an interesting point that we've now worked on quite a bit more, and we're really excited about, is trying to understand the sort of the more holistic effects of cattle, where cattle behavior or cattle activity on the landscape is determined by human regulation in the villages themselves.
Right.
Some villages will go out of their way to actually clear the organic rich sediments out of these water bodies when they dry at certain times of the year to remove all this excess cattle manure.
So you go into this project thinking these hybrids are bad. This is a risk that, we really need to get our hands on. Then you find there's hardly any hybrids, but you discover that the cattle that you thought were important for the hybrids are bad because they defecate in the waters. And, can really sustain these snails and thereby drive these parasites and epidemics potentially. So, but then what I like about it is that it seems that there could be some really straightforward solutions. Like you said, there are some water bodies where you say cattle cannot go there, so we separate the cattle from the people. Is that too simplistic, or would that be one way to
I think that's
control this?
I think that's one really great way, and we're actually thinking about initiating those kinds of field intervention trials now. And so we're thinking about combining those kinds of behaviors, like really reinforcing restriction, but also thinking about landscape modifications that can assist with blocking, say, runoff from bringing cattle manure into water bodies. So some of the water bodies are surrounded by raised walls that have been built. So combining fences and raised walls, which are already elements on the landscape, could be really good. But we also think that there are really, really critical ecological feedbacks. So there's some work done from a branch of ecology called critical transitions theory. And what this research says is that you can stop all these nutrient inputs, but recovery from eutrophication takes a long time. And one of the potential reasons is that organic material, all those nutrients, have accumulated in the sediment. And the sediment provides a slow release of nutrients back into the system. And we've recently hypothesized that, building these barriers or reinforcing these regulations might not be enough without that human intervention of removing the, putting a brake on that positive feedback in the system. And so that's where we're really excited to move next, is try to think, how do you actually implement these kinds of interventions that really take into account the human behavior, the needs of an agricultural community, and the ecological feedbacks that might exist.
Well, this has been very interesting research. I wish you a lot of luck with your ongoing projects. We hope you enjoyed this episode and look forward to seeing you for the next ones. Thank you for watching.